All right, in this video we are going to start the chapter on cells. And so first we're going to do an overview kind of cells, and then we'll talk about the membrane, and then we'll move inside of the cell and start talking about some of the different organelles found in the cell. Okay, so overview of the cell. These are things that are present in almost all cells. Okay, so common structures. All right, so the first one is the cell membrane. Okay, so this would be the outside of the cell. All right, so this outer edge, it provides the barrier around the outside of the cell. That's the cell membrane. We talked about last time, it's made up of phospholipids. And then the nucleus of the cell is going to be in the center. All right, or actually it doesn't necessarily have to be in the center, just is on this cell. And that's going to contain the DNA. So most, not necessarily all cells, but most have a nucleus. All right, some even have more than one nucleus. And then we have the cytoplasm, which would be kind of this tannish white area all around the nucleus. Okay, and so the cytoplasm consists of the cytosol, all right, which is like the kind of jelly goo that makes up the inside of the cell, and all of the organelles that are suspended in it. Okay, so a lot of times cytoplasm is used when someone actually means cytosol. So cytosol is the actual kind of fluid inside of the cell, whereas the cytoplasm includes that and all of the organelles we're going to talk about. Okay, so basically everything from the nucleus to the outer edge of the cell to the membrane, that is all cytoplasm. All right, so now let's talk about the cell membrane and its structure. Okay, so we said last time the cell membrane, all right, is made up of phospholipids. Its function is to provide a boundary between the outside and the inside of the cell. So intracellular fluid, this would be on the inside of the cell. Okay, and then extracellular fluid, that would be on the outside of the cell. And we're going to learn in some of the different chapters that we go through in A&P 1. There are some really important things that are very, very different between the inside of the cell and the outside of the cell. So for example, sodium, right, is going to be very different inside versus outside of the cell. So outside of the cell, we're going to have a very high concentration of sodium. And then the inside of the cell is actually going to have a very high concentration of potassium. And that is going to be critical for nervous system function and for muscle contraction. All right, so we'll talk a lot about those when we get to those chapters. Okay, another function of the cell membrane is that it helps to identify the cell. All right, and we'll talk about that when we talk about some of these membrane proteins. There are proteins and sugars on the surface of the cell that help to identify the cell as belonging to self. All right, so you know if you give somebody like a transplant or a transfusion of blood, it has to be a match, and that's because your immune system is looking for these identifying markers in the plasma membrane that identify a cell as belonging to you, right? So being of you, being a self cell. Okay. And then there are also some structures in the membrane that allow communication between cells. Okay, so they can talk to each other. So there's some kind of receptors and that kind of thing that can interact with other cells. All right, so let's talk about that phospholipid bilayer. All right, so with the phospholipid bilayer, it is made up of, so bilayer meaning it has two layers, so bi meaning two layers of phospholipids. All right, so this would be one phospholipid here. All right, this would be a second phospholipid here. And we know with our phospholipids, they have two different regions. They have a polar region that is going to face to the outside and the inside of the cell. All right, that polar is going to be hydrophilic. right? It likes water. Okay, so hydrophilic, this is the polar head of that phospholipid. All right, so this is going to face both the inside, that intracellular fluid, and the outside of the cell, because both of those areas are made of primarily water, and so you want something that is polar to face each of those areas. Okay, and so then in the center here, we have a collection of our fatty acid tails. All right, those are going to be hydrophobic, meaning they don't like water because they are made of fats, right? So these are going to be the fatty acids. Okay, so they're going to make up this kind of core, right? This inner layer that's a core going through that phospholipid bilayer. And this is actually what's going to provide that barrier that helps to keep the outside different from the inside. So polar things can't just, water itself can't just move through the membrane, all right, inside or out. Ions can't just move through the membrane however they want to, all right? So you keep a separate environment on the inside and the outside. Okay, so only non-polar things would be able to pass through this inner hydrophobic core of the phospholipid bilayer. All right. Cholesterol. So the membrane does have cholesterol embedded in it, and cholesterol, so that would be these guys. Whoops. I'll switch to, there we go. All right, these guys here are going to be cholesterol. They're going to help to make the membrane more rigid. And so this can be different just depending on the cell type. Sorry, that was my dog coughing. So this would be different. So different cells would have different amounts of cholesterol just depending on what they need. So they determine how rigid or flexible that cell membrane is. Okay, so cells that crawl around a lot, right, move around, might not want as rigid of a cell membrane as some others. Okay, so now let's talk about membrane proteins. So within this phospholipid bilayer, there's going to be embedded a lot of different proteins with a lot of different functions. All right, so if a protein travels completely through both layers of the bilayer, it's called either a transmembrane, right? Trans because it spans the entire membrane, or an integral because it's completely integrated into the membrane, okay? So transmembrane protein or integral membrane protein, okay? And so these are going to be through both layers of that phospholipid bilayer. And those are actually referred to as leaflets, right? So both layers or leaflets of the membrane. And you could also have proteins, right, like this one down here, that just sit in one layer of the membrane. That is called a peripheral membrane protein, okay? So these are going to be in one layer. And so it doesn't matter, well, it does, it depends on the protein, so of the membrane, right? So whether it's on the layer that's closest to the outside of the cell or the inside, that would just depend on the protein and its function, all right? And so an example of this would be sometimes there are enzymes, right, that would be peripheral membrane proteins, and they can sit on the inside of the cell and catalyze different reactions right there near the membrane. Okay, so let's see, our first specific kind of integral membrane protein is a channel protein, okay? And so channel proteins are going to be really, really important when we talk about the nervous system and muscle contraction. Okay, and so channel proteins allow movement, right, through that channel. Typically, they're going to be specific for certain types of things, all right? So allow movement through the membrane. Okay. All right, and so this movement is just going to be from high to low concentration. All right, so from high to low concentration. right? And that's specific for channels. So they allow movement of different molecules from high to low concentration. One good example of this would be sodium, right? So we said there's a lot of sodium outside the cell, right? So we'll put a big sodium there. We'll put a teeny tiny little sodium here because there's not a lot on the inside of the cell. And so if this were a sodium channel, it would allow sodium to move from where there's a lot on the outside, right, across the membrane to where there's a little on the inside. Okay? All right, so that's channels. They allow movement from high to low concentration. There are also transporters. Okay, and so transporters allow movement through the membrane, all right? Okay, so we'll just put that again here. Allow movement, we'll just say through. However, it's going to be a little different. So with transporters, they're going to be going typically from high or from low to high concentration. So a transporter is going to actually grab a molecule and push it to where there's already a bunch of those molecules. So it's not just like a hollow tube that allows something to move through, like a slide, right? It's physically going to grab a hold of something, all right? So what it would do is this one would start kind of open, right, to this side. It would grab a hold of these little, balls on this side, right? They would bind, and then it would flip and open to the other side like it is now and release those little molecules on the other side. And so an example of this would be glucose, right? So if you're absorbing glucose, it would be grabbed here, right, and it would be moved into here where there will do big glucose is already a lot of glucose. Okay, so it's going to go from low to high concentration. Okay, and so that's typical, okay, for a transporter. There are some transporters that work a little bit differently that when we encounter them, we can talk about them. But kind of typically as a major category, that's kind of how transporters work, okay? All right, and then lastly, we have an example of a peripheral membrane protein. So this would be a glycoprotein, okay? And so this would be an example of the type of proteins that are used to identify the cell, right? So these ID the cell. Okay, and there actually can also be the lipids, the phospholipids themselves can actually be kind of decorated with sugars. Those are called glycolipids, all right? And those can identify the cell as well. So glycolipids and glycoproteins do similar things. So just adding sugars on the outside of the cell, whether you're adding it to a protein or to one of the phospholipids, those are going to be ways to identify the cell. A really good example of this would be on red blood cells. So red blood cells, we can identify them as being either type A, B, or O. Those identifying markers are actually going to be these based on these surface sugars, okay? Whether they have type A sugars on their surface or type B or none at all would be type O, okay? All right, so that is the membrane proteins, right? The different possible ones, and we'll talk about some examples of them as we go throughout the semester. Okay, so let's talk about junctions because this does involve also interactions between kind of the cell membrane between cells. And then we can move on to other organelles. Okay, so with cell junctions, there's three types we're going to talk about. So the first type of cell junction is called a gap junction. Okay, so gap junction is where an actual tube, right, a channel forms between cells. Okay, and so gap junctions allow direct communication between cells. Okay, So they have like a direct line to each other. And we see this, one really clear example, we see this is with cardiac muscle cells. All right, so we can make a note of that here. So cardiac, so heart muscle cells, have gap junctions between them. And so that's because when one cell starts to contract, right, it gets that electrical stimulus to contract, you want to be able to really, really quickly pass that information, that stimulus, from one contracting cell to the next, so that the whole heart kind of contracts as a unit. Okay, so it allows for really fast communication between cells. Okay. And then we have tight junctions. So I always think of tight junctions as being watertight, right? So think of like a ziplock between cells, right? So these are watertight. Okay, and so these are places where you want to make sure fluid cannot move between the cells. So a good example of this would be the skin. You definitely don't want, water being able to seep out of your skin. The stomach, right? You certainly don't want stomach acid being able to seep out of the stomach, okay? And so you'll have tight junctions between those cells. All right, and then our last type of junction is called a desmosome. All right, so these are going to be kind of like anchors. They're really, really strong and tough. They resist tearing and stretch. All right, so also you can think of it as kind of like welded connections between cells. All right, so anything that would undergo a lot of stretch and pulling, again, the skin would be a good example of this. And actually heart as well would have these, right? So cardiac muscle, because that does undergo a lot of stress, right? The heart contracting, it never stops, right? So cardiac muscle would definitely have those. And as you can see, cells can have combinations of these three. So cardiac muscle, they have special junctions between cells that you're going to learn about called intercalated discs, which consist of both gap junctions and desmosomes. Okay, so you can mix these, just depending on the needs of the cell, you can mix these different junctions together. All right, so now let's move into the cell and start talking about some organelles. Okay, and so as we go through organelles, we'll kind of point to them on this picture and label them and then talk about functions and that kind of thing. Okay. All right, so within the cell, we can have inclusions. And so inclusions are just little kind of pockets, right? Clusters of fats, pigments, and glycogen, right? Remember from our last chapter, glycogen is the stored form of glucose. And so you could just have some little pockets of those within the cell, depending on the type of cell, right? You might have more of one than the other. Okay, and then when we talk about actual organelles, we're going to say they're either membranous or they are non-membranous. Okay, so a membranous organelle is going to be surrounded by phospholipid bilayer, a separate phospholipid bilayer from the one that creates the outside of the cell. Right, so it's going to have its own phospholipid bilayer. All right. And then there are non-membranous organelles, which do not have phospholipid bilayer. So, example of this would be a ribosome. All right, it's a non-membranous organelle. Most of the organelles you probably are already familiar with are going to be membranous. Mitochondria, the nucleus, the endoplasmic reticulum, all of those are going to be membranous. Okay? All right, so we're going to start off talking about the nucleus, okay? And so the nucleus is kind of like the brain of the cell. It's where the blueprints for building cells is located. The Some cells can be multinucleated, right? That means many nuclei. So that would be plural, nuclei, plural of nucleus. Some examples of this, one example you're going to learn actually in our next chapter when we do tissues is going to be muscle. So skeletal muscle, right? That's going to be the type of muscle that, you know, is in like your biceps, right? That you can control voluntarily. That's called skeletal muscle. They are multinucleated. And then there are some cells that are anucleated. That means they have no nucleus at all. An example of that would be a red blood cell. So red blood cells don't have a nucleus, okay? All right, so then when we look at the nucleus, it has kind of an outer layer, right? So if this is our nucleus, that outer layer is called the nuclear envelope, okay? And so the nuclear envelope is a double layer of phospholipid bilayer, right? So it's a double membrane. All right, and so that means that it has two complete layers. So this would be phospholipid bilayer, and then this would be another layer of phospholipid bilayer. So going all the way around the nucleus, it has two sets of phospholipid bilayer. So it has a double membrane, and it has pores. All right, and so those pores are going to be really important, right? So these are the pores here, these little darker spots. Okay, so the pores are going to be important for movement into and out of the nucleus. All right, and so we know that in the nucleus, that's where the DNA is found, all right? And so you need to be able to make a copy of that DNA and send it out of the nucleus. And so that's a really important function of these pores, being able to send kind of the cellular machinery in there to get that copy and then take it back out. Okay. So then inside of the nucleus itself, we're going to have the chromatin. So that's these kind of thread-looking structures. And so chromatin is going to be uncondensed, right, DNA. And so DNA, when it's kind of in this DNA spaghetti form like we have drawn in this nucleus, right, it's not condensed, that is called chromatin, okay? And so that DNA is packed around these packaging proteins called histones, all right? So histones, that's what these guys are. So the DNA is just the blue thread. It's wrapped around these histones. These histones are made of protein, all right? And they're just going to help to organize the DNA. That's just its job, all right? If you think about this DNA, you know, being like this big thing of spaghetti, right? You want to kind of be able to, you know, make sure it doesn't get all tangled up, organize it a little, and when you want to make a copy of that DNA, you can easily find that spot you want to make a copy of. So that's what the histones help with. All right, so when the DNA condenses, and the only time it'll do this is when the cell is about to divide, all right, it is called a chromosome. It condenses into a chromosome, all right, condensed DNA, and that's going to be prior to cell division. Okay, and so you guys have probably seen pictures of chromosomes, right? They're usually drawn as kind of this X structure, right? Like this. Okay, and then these guys would be connected in the middle like that. So that's typically how a chromosome is drawn, all right? So we'll talk about, when we talk about cell division, right, in our mitosis chapter, our mitosis section, we'll talk about those chromosomes and the different parts of them. Okay, but those chromosomes only form when the cell is about to divide and split into two new cells. Okay, and lastly, within our nucleus, cell division, there we go, we have a nucleolus. And I swear, this is the hardest thing for people to remember what the nucleolus does. The function of the nucleolus is to make ribosomes. All right, so that is the job of the nucleolus. That little dot within the nucleus, its job is to make ribosomes. Okay, and we'll talk about ribosomes. They're just these little purple dots all throughout the cell. Their function is to make proteins, but those ribosomes are made in the nucleolus. All right, so let's just talk about ribosomes really quick. Okay, so ribosomes, as you can see on this picture, they can be either fixed or they can be free. All right, so a free ribosome is floating in the cytosol. All right, so floating in the cytosol, and then a fixed ribosome is going to be attached to rough ER. All right, so it's going to be attached to endoplasmic reticulum, which we're going to talk about in the next video. All right, so endoplasmic reticulum. Okay. Yeah, okay, we are in the next video. Yeah. So ribosomes, their job for both of them, whether they're fixed or they're free, is to make proteins. Okay, so they make proteins. What's going to be different between the two is the type of protein made by each. All right, so fixed ribosome, right, these ribosomes out here that are just floating around in the cytosol, they would make proteins that are also just going to float around in the cytosol. Okay. However, these fixed ribosomes here that are attached to the endoplasmic reticulum, they're going to make proteins that they're going to be made into this little pocket in the endoplasmic reticulum, all right? And those will be packaged up in these little bubbles, and they can be sent elsewhere, all right? So they can be sent, to other organelles, right? They can be sent out of the cell if you have a protein. Maybe you're making like a protein-based hormone that you want to send out of the cell, right? You can do that. So the protein is what's different between these two types of ribosomes. So free makes kind of free proteins that float in the cytosol. Fixed makes proteins that are going to be kind of packaged up and sent either somewhere in the cell or outside of the cell. Okay? But the job of a ribosome is to make proteins. Okay, so let's talk just very briefly about protein synthesis. Okay, and so with protein synthesis, that DNA, right? So making proteins, right? So the DNA, we know it, so it cannot leave the nucleus. All right, so the DNA, in order to get the information on the DNA out of the nucleus, it has to be copied. All right, so it's copied into what's called mRNA, messenger RNA. All right, that messenger RNA leaves the nucleus. Okay, and then it goes to find a ribosome. All right, so it goes to a ribosome. It says, here are the instructions for making a protein. You need to put these amino acids together in this order, right? This is the primary structure for that protein. So that ribosome is then going to assemble the amino acids, right? And it actually is able to form, so it forms that peptide bond we talked about in the last chapter. All right, so there's an enzyme inside of that ribosome that can form that peptide bond. Okay, so that's kind of generally how proteins are made. So the DNA, if we're looking at the cell here, right, and I'll leave that so we can look at it. So the DNA here, we're going to need to make a copy of it, right? So we make a little copy. So we have this little copy thread that's going to travel out, right, through a nuclear pore. It's going to go find a ribosome, say, here's my blueprint for how to make a protein, give it to that ribosome, and that ribosome is going to start putting together amino acids in the order described by that messenger RNA, and then that will form into our protein. Okay, whoops, didn't finish this word. I got too busy talking. Forms peptide bond. There we go. All right. And so then the last thing we're going to talk about in this video is going to be the mitochondria. All right, so this is typically the organelle that everybody already knows at least a little something about. I'm sure if I asked you what is the function of the mitochondria, you would tell me that it is the powerhouse of the cell. All right, because that's what everybody learns, in high school or general biology class. We're going to go a little further than that. So I don't want to see powerhouse of the cell on your test, right? If you're asked what the function of mitochondria is, it is to make ATP. All right, so that ATP is the form of energy. We talked about that in the last chapter too, used by the cell. So it is essentially, if you boil it down, the powerhouse of the cell. But I want you to be specific. I want you to tell me that the mitochondria makes ATP, okay? And so it does that through a process called cellular respiration. And we're not going to go into detail this semester about cellular respiration, but you'll learn about it with metabolism next semester, okay? And so the structure of mitochondria is also unique. Similar to the nucleus, it has a double membrane, right? That means it has two layers of phospholipid bilayer. All right, so it has this outer layer out here, right? And then it has this inner layer here that's kind of squiggly like this. Okay, so two layers of phospholipid bilayer. All right, and so there's two structures you should know with the mitochondria. So this wavy inner membrane, right, forms these kind of little peaks like this here, right? These are called cristae. Okay, cristae are where the last step of cellular respiration happens. And then within the middle of that, all this orange area in the middle is called the matrix of the mitochondria. In that matrix, we're going to have a bunch of stuff like enzymes for cellular respiration. It actually contains DNA. Mitochondria has its own DNA, all right, that's separate from the nucleus. Okay, and so we would also have ribosomes in there. If you have DNA, you need to be able to make proteins. So we can say own DNA and ribosomes. All right, so mitochondria is a really unique organelle within a cell. Okay. All right, so we're going to stop there, and then we'll pick up with the smooth endoplasmic reticulum in our next